So I just want to start off by introducing this webinar series. So within the European Joint Program on Rare Diseases, the work focus group called Resources for Sharing Experimental Data and Materials has scheduled a series of monthly webinars throughout 2021. And these seminars aim to introduce each resource to the rare disease community. So I would encourage you to check out our schedule of upcoming seminars at the web address at the bottom of the screen. And yeah, today is my turn, June 21st. So today I will present hPSCreg. So today I'm going to give you a short background on the human pluripotent stem cells, a short overview of the registry itself, and then I'm going to present three main resources in hPSCreg, including cell line data, the clinical study database, and research projects. So I hope to also highlight the utility of hPSCreg resources for stakeholders from research, industry, and funding agencies. Okay, so pluripotent stem cells are cells that can form almost all cell types in the body. Embryonic stem cells can be isolated from the inner cell mass of embryos, and induced pluripotent stem cells may be generated from somatic cell types, such as skin cells or blood cells, by a process called reprogramming. Reprogramming can be carried out by overexpressing the so-called Yamanaka factors, and these are transcription factors OCT4, SOX2, KLF4, and MYC. These pluripotent stem cells have the advantages that they are a renewable source of cells. You can use them to generate specific cell types that may be difficult to obtain, such as brain cells. And any experiments that you perform would be based on human biology rather than an animal model, and this could possibly reduce the need for animal experimentation. There are many potential applications of human pluripotent stem cells. They are versatile reagents for generating disease models for drug discovery. The cells can be genetically modified to normalize or introduce mutations and then derived to make specific cell types, which can be used in a variety of technologies, such as organoids, microfluidic systems to model cell-cell interactions and flow, 3D printing of cells. And specific cell types can be generated for cell replacement therapy, for example, by repopulating biological scaffolds with cells and then transplanting them into patients. Human Pluripotent Stem Cell Registry hosts a number of sources online, including a database for pluripotent stem cell data, research projects using registered lines, and clinical studies that use stem cell derived cell types for therapy. We also host the, we also host the stem cell data for the European Bank of Induced Pluripotent Stem Cells. This is a biobank of iPS cells. And in the future, let's see, so within, for the eBISC biobank, we have 896 iPS cell lines available, and we host all of this data. And in the future, we will also host the data for the iPSC derived cell products being produced by eBISC. Okay. Yeah. Right, okay. Okay, so in the next few slides, or for the remainder of the talk, I will talk about the three main resources. Our biggest resource, of course, is the cell line data. And then I'll talk about the research project database, and finally, the clinical study database. Okay. So For the cell line data, where does this cell line data come from? It actually comes from you, the research community, and we encourage you all to register your pluripotent stem cell lines in hPSCreg. So what are some of the benefits of registering your lines? First of all, you can increase the global visibility of your research. hPSCreg contains cell lines from over 40 countries in the world. Secondly, you can make your cell lines more accessible and findable. Because when you register your lines, we will assign them a unique reference code, which will allow your cell line to be traced throughout research and projects. We also use biosamples identifiers from EBI. If you enter all of your cell line data at hPSCreg, you can publish this as a stem cell research lab resource. And finally, registering your line promotes data transparency and ethics compliance and pluripotency evidence. So if you provide the required information for ethics and pluripotency, you may be eligible to receive a certificate from hPSCreg, which attests to the ethical provenance and biological properties of the line. And these certificates are recognized by the EU for EU-funded research projects. So in the next 2 slides, I'm going to introduce the kinds of data we collect for the lines. First, as I mentioned, we assign a unique reference code, and this is outlined here. It's composed of 5 different parts. The first part is an institutional acronym. Second part is the type of pluripotent stem cell, so either induced pluripotent stem cell or an embryonic stem cell. There's a running number for the donor from this institution, and the individual lines from this particular donor are then further annotated by ABCD, etc. And finally, a subclone is donated by a suffix at the end, -1 or -number. So within hPSCreg, what we mean by subclone is any genetically modified pluripotent stem cell line. For example, if a line contains a reporter gene construct or has been engineered so that it introduces a mutation or normalizes a mutation, it would become a subclone in our system. So as an example, you can see here for the University Clinic Bonn, UKB, this is apparent from the institutional acronym. It is a induced pluripotent stem cell line. This first line from the first donor and the first line from the donor, and there's a subclone, so it has been genetically modified. So at a glance, using these identifiers, you can get a little bit of information about the stem cell line. Okay, so in this slide, these are all the fields that we collect for the registration of a cell line. The fields that are outlined in this pink color are currently mandatory fields, and this means that these fields must be filled before your cell line data becomes public on hPSCreg. These mandatory fields include information about the generator, so who made this line. It's usually an institution who fills this out. Information about the donor. We also use the biosamples donor ID, any diseases associated or diagnosed with the donor, what is the genetic sex of the donor, and we also collect a vast amount of data on the ethics and consent surrounding the donation of this material for iPS lines or for ESC lines. For the cell line data, we have information about its derivation, either as an embryonic stem cell or as an iPS line. We also record information used to characterize the pluripotent stem cells, such as undifferentiated stem cell markers, and information about its pluripotency, in particular, its potency to form the 3 germ cell layers, the 3 germ layers. So I'm not going to describe all of the data in detail. If you are interested in registering your lines and would like to find out more, you can contact us directly. So in the next part, I would like to focus on using the data we have in hPSCreg. And in particular, I'd like to focus on rare disease users. So how can hPSCreg be useful for the rare disease user? Oh, I just want to point out that we are now an ERDC recognized resource. So hopefully that will give us more traffic for the rare disease community. Okay. So you might ask, what is the rare disease content in hPSCreg and how can I use hPSCreg? And I will try to explain this in two examples of a search. One is just going to be a simple search for lines, and the second is a more detailed search. So while hPSCreg is not a rare disease specific resource, approximately 21% of the lines that are registered in hPSCreg come from donors with a rare disease. Looking at the total number of diseases represented in hPSCreg, rare diseases account for about 55% of all diseases associated to lines in hPSCreg. So although rare disease is individually rare, they are collectively common. Okay, so in our first example of a search, we're going to look for lines that are derived from donors with Brugada Syndrome. So here you can see the entry for Brugada Syndrome from the Rare Disease Resource Orphanet. Orphanet is the portal for rare diseases and orphan drugs, and you can learn more about Orphanet in their webinar later this year. So Brugada Syndrome is a cardiac disorder characterized by abnormal electrical activity in the heart. It is diagnosed by electrocardiogram, and there is a genetic component. There are some genetic variants that are associated with Brugada, including these ion channels or proteins that interact with these ion channels. There are treatment options, but there is no cure for Brugada syndrome. So if we are to search for Brugada syndrome lines in hPSCreg, we can enter Brugada in the search window. And this will yield a list of 7 lines that have been associated with Brugada syndrome. These are all hPSC lines. One is from Leiden University Medical Center, and the rest are from the University Clinic in Cologne. And you can see from the cell line names, there are 3 cell lines, A, B, and C, from a single donor, which is 024 from the University Clinic of Cologne. Yeah, so there are 2 donors from University Clinic of Cologne, each with 3 lines from these donors. Now, if we click on the cell line record of 1 line, we can see this is what we have some general information about the name, alternative names, who generated the line. In here, we can see that this line is being distributed by eBISC, and it is also kept by the European Collection of Authenticated Cell Cultures, or ECAC. We also have a number of links to external databases, including Biosamples and Cellosaurus, which is another resource that was presented in this webinar series. Here we see it's associated with eBISC, the European Bank of Induced Pluripotent Stem Cells. And here, this is a very interesting information, shows you the availability of this cell line for research, clinical, or commercial use. And then we have like other information about the donor. It was a male, age was between 65 and 69. And in particular, this donor has been genotyped for a particular mutation in the SCN5A gene. And if we scroll down through the stem cell record, you can see what kinds of Marker expression was used to characterize its undifferentiated stem cell status. There's some pictures above showing the cells. And finally, the results of microbiology and virus screening was all negative for HIV 1, 2, hepatitis B, C, and mycoplasma. And finally, at the end, we see that there's some genotyping data available for this cell line. And it looks like it was an SMP array. So now, if we're interested in seeing where we can actually obtain this cell line, we can click on purchase this cart to go to eBISC. This brings us to the catalog entry for this line at eBISC. And it has a lot of the basic information that we've already seen. And as well, you can click on this shopping cart to go to ECAC to buy this line. But that's not all. You can also look for other Brugada lines at eBISC by going through their search, performing a search in the catalog. So you can click on Brugada Syndrome here, and there are indeed 6 lines, and they're all from the University Clinic of Cologne. So these lines are available for purchase at the eBISC catalog. Okay, so just to summarize what we've done with this simple search, we found 7 hPSC lines from 3 donors. 6 of these lines are biobanked at eBISC, so they're readily distributed. And I didn't go through all the lines, but if you were to go through all the lines, you would find out that the UKK lines from the University Clinic of Cologne are available maximally for research, clinical, and commercial use. And in particular, one of these lines from one donor has a genetic variant which is associated with Brugada syndrome. Okay, so in my second example, oh, no, wait. I just want to say something about eBISC. So this is a biobank of induced pluripotent stem cell lines. It has 185 lines that are associated with rare disease, and this accounts for about 20% of the lines that are banked in eBISC, and you can see a complete list of all the lines and diseases here. And if you would really like to know more about eBISC and are attending the ISSCR annual meeting, which coincidentally starts today, you can view the focus session on eBISC, and it starts at 7 p.m. today. Okay, so next I will do my second example query, which is going to be a little bit more complicated. So let's say we want to search for lines with a lot of different properties. For example, I want to collect a cohort of lines from a group of related diseases. I want to sequence them, generate high throughput organoids on a microfluidic device, and sell it as a commercialized panel for drug screening. So what are we looking for here? We want pluripotent stem cell lines from the same disease. We want some kind of sequencing data if it's available, and commercialization must be allowed for these lines. So for this example, I've just chosen Alport Syndrome. So Alport Syndrome is a rare renal disease, and here's the entry from Orphanet once again. And it's a chronic kidney disease and results in hearing loss. Oh, diagnosis is not by ECG, that's something else. Genetic variants in this disease are usually the type 4 collagens. And as for the treatment options, all one can do is delay the onset of end-stage renal disease and then ultimately get a kidney transplant. So how do we go about finding lines for Alport syndrome? First of all, we can go to the website and select diseases under the browse menu, then enter the term Alport. And this will list all the diseases that are associated, or sorry, all the cell lines that are associated with Alport syndrome. In particular, we have 2 Alport syndrome entries. And if you have recognized how the stem cell lines are named, you will recognize that this one is actually a subclone because of the suffix -58. So this is actually a subclone that has been genetically modified to contain a genetic variant associated with Alport syndrome. For all of these lines here, there's no subclones in them. So these are all lines that have been derived from donors with Alport syndrome, and these are 19 lines. So you can select one line. I've just picked this line here from, it's called WTS I519-B. And we can look at the cell line record here. Again, a lot of general information. It's available for purchase at ECAC. It's not an EBIS line in this case. There are links to external databases such as Cellusaurus. Oh, and particularly here, the availability is apparently limited to research. Disease was diagnosed, and if you go near to the end of the record, you can see that there is some genome-wide data on the cell line. In particular, there's some exome sequencing and RNA-seq sequencing data, and this is available at EGA, which is incidentally also another resource that you can see on our webinar series. So what have we found out? There are some lines from Alport syndrome, 19 of them. At least some of them have exome sequencing data. However, commercialization is not allowed. And so just to summarize, I'm not going to go through all of the lines in detail, but we have 19 lines associated with Alport syndrome from 12 donors. 9 of these lines have exome sequencing. Unfortunately, commercialization is not allowed. It's only allowed for research. And at least 15 of these lines are distributed by ECAC, so they are readily available for purchase. Okay, so although we were not successful in our search to find lines for commercial application, I hope this demonstrates to you how you can find different kinds of information using the hPSCreg website. Okay, in the next portion, I would like to present the research projects. So we have 204 research projects registered in hPSCreg. And let's see. Yeah, so if you click on the Browse menu Research Projects, it will lead you to this page, which gives you a list of research projects. Most of these projects are from, they are EU-funded projects. Most of them are from the European Research Council, Framework Program 7, Horizon 2020, with 26 projects. Then that includes the Maria Slodska-Curie Action Projects, which constitute 18 Marie Curie projects. And there are other funding agencies associated with some of these projects. In particular, the Joint Transnational Call for Rare Disease Research projects, I was able to look at the 2018 and 2020 calls for the JTC, and I found that over 50% of these projects use IPS. So this is quite promising for us. So at this point, funders can use this resource to monitor research outputs in terms of cell lines and how many projects are using the same cell lines. And industry and researchers can maybe use this resource to find collaboration partners with a particular kind of expertise. Okay, so in the final resource I present today, this will be the clinical studies database There are 82 clinical studies, and you can access this by going on Browse and scrolling down to clinical studies. So the motivation for providing this particular resource is, or are, reliable data about stem cell therapies is difficult to find. So you're probably aware of a lot of unproven stem cell clinics offering miracle treatments to the highest bidder, basically. We also include in this database studies that specifically use human pluripotent derived cell types for cell therapy. So this does not include mesenchymal stem cells or other adult stem cells. This is only about pluripotent stem cell products. And we hope that this will enable the tracking of stem cell lines from research to clinical application. So in the next slide, this is a little bit complicated, but this is a Sankey plot showing the starting cell line types, either embryonic stem cell type or the induced pluripotent stem cell types, and showing their usage towards a clinical application. So from this column, you can already see that the majority of clinical studies use embryonic stem cells. And as for the derived cell types from these stem cells, it's a lot of retinal pigmented epithelia. And these retinal RPE cells are used for clinical applications in macular degeneration. Okay, so these numbers are a little bit old, and I'm sorry I haven't had time to update this, but it just gives you a sense of what kinds of pluripotent stem cell types are being used for what end applications. Okay. Looking worldwide, clinical trials are taking place most numerously in the US and in China, followed by Japan, Great Britain, the Republic of Korea, South Korea, that is, and Australia and Canada with 4 each. Okay, so that's just a really quick overview of the clinical trials which involve human pluripotent stem cells. So in the last, so I'm just going to summarize now quickly. So I've shown you today all the resources that are in hPSCreg that you can access. These include the cell line data, for which we issue certificates to document the ethical provenance and biological properties of stem cells. We have the research project database, and finally the clinical study database. For future developments, we want to perform more verification of the data, and this would include using data use ontology and informed consent ontology with regard to the ethics surrounding the donation. And as I was making this presentation, I realized that we could actually collect a lot more data on the research project. So we hope to associate these research projects with a better annotation. We are going to develop use cases with other European rare disease resources to increase the search capabilities with, for example, RD Connect and InfraFrontier. And if you have any other suggestions, please let us know. We are going to be expanding the hPSCreg knowledge base to so-called regulatory primed stem cell lines. So these are lines, pluripotent stem cell lines that are being prepared for clinical translation. And we also want to expand our database to include human pluripotent stem cell derived cell products. And as a last thing, we are prioritized for integration into the EJP RD virtual platform this year. So I would like to acknowledge the hPSCreg team and our advisors for the ethical and scientific part. We also recently moved from the Charité to the Fraunhofer, and now we are located at the Fraunhofer Institute for Biomedical Engineering with an office in Berlin, Germany. If you would like to contact us, you can reach us using our contact form